Good afternoon, everyone. Before we start the session, I'll acknowledge the country first. So on behalf of TRI, I pay our respects to the Tribal and Jagera people, the traditional custodians of the land and waters of the Brisbane area. We recognize their continuing connection to land, water, and culture. We pay respects to elders past, present, and emerging. We acknowledge the stories, traditions, and living cultures of Aboriginal and Torres Strait Islanders people, and commit to fostering a culture of learning from and working with First Nations people in the spirit of reconciliation and access to justice. So welcome everyone. My name is Kavita Bisht, and I'll be chairing the session today, which is for Research Translation Seminar, focusing on translating your therapeutic research to clinical trial. So today we have 4 fantastic speakers. So I will introduce them one by one, and we will have Q&A towards the end. So our first speakers are from 360 Biolab, Angela Luttick and Derek Richard. So Angela is a co-founder and executive vice president, commercial at 360 Biolabs, and leads business development and commercialization strategies for the company. Angela is a virologist with more than 20 years experience in the discovery, development, and commercialization of novel drugs. And Angela has very well experience in the generation and integrity of key discovery stage biological data required for the preparation of IND packages used in filling numerous FDA submissions. So please welcome Angela.
Hi, everyone, and thanks for your attendance today. I'm going to present a very exciting time for us at 360, which was, March about 2020, when COVID just hit. And when you're in the clinical trial space and you have no idea whether that means that you can continue supporting clinical trials or whether, clients are even going to want to conduct clinical trials in Australia, we did what we call is a little bit of a flip. We used to have an R&D team that we closed down in 2017 and moved purely and focused on clinical trials. And what we're seeing is a lot of interest in some more R&D type experiments and work once COVID hit. So we opened back up our R&D team. And we had a great experience working with the CARP team for one of their innovations and one of their testings against SARS-CoV-2. So I'm just going to introduce 360 Biolabs a little bit because I'm not sure if you know what we actually do, but then we'll segue across to Derek, who will present the work that we did together. So 360 Biolabs is a specialty lab, probably the most comprehensive specialty lab in APAC. So we have an industry experienced executive team. I led a biology team for 15 years at Biota Pharmaceuticals prior to starting 360 Biolabs. So we've been and we've walked in our clients' shoes when it comes to taking products towards the clinic. Our laboratory does PK and PD all in the one lab. We're able to develop client-specific assays that aren't necessarily available elsewhere. We have internationally recognized quality systems, GLP, ISO, and GCP. All of our assay validation and reporting is compliant with FDA because a large component of our clientele is US-based. And we pride ourselves on delivery of high quality data on time, every time. So just going into a little bit of the science, we have a small molecule bioanalytical team that does proteins, peptides, as well as the small molecules, a range of different adjuvant and SciEx instrumentation. We also support large molecules. We could obviously do ELISA builds as well as MSDs. And then when it comes to our pharmacodynamic endpoints, we have an immunology team, a virology team, a molecular team. And so we can support really any endpoints that come for your clinical trial. We do a lot of flow, a lot of biomarkers, a lot of ADA. The virology team has been extremely busy over the last 2 years. We were lucky enough to grab the virus from the Vigil team in February of 2020, grow the virus and work it into some microneutralization assays. And we supported Novavax that came to do their first phase 1 clinical trial in Australia, and we're still supporting them through to phase 3. So I think we've just supported about 25 different COVID vaccine clinical trials over the last 2 years. As I mentioned, we do have a molecular biology team, so we can do a wide range of molecular endpoints for clinical trials, and also central lab services. So lab manuals, site training, sample collection kits, and all the things that you need to actually collect the samples to do the analysis on for a clinical trial. And we support a wide range. I don't know if you're aware, but you know, clinical trials in Australia are very, very diverse. There's a wide range of different biotech companies and pharmaceutical companies across the globe that come and conduct their clinical trials in Australia. And so you basically just has to have a lab that can support a wide range of PD endpoints and develop assays that's specific for a particular molecule or a therapeutic area. So in March 2020, Victoria was declared a state of emergency. It was the first of our lockdowns. We went through 6, as you're all aware of, and it was a very, very difficult time. As I mentioned before, it was hard to know whether the business was going to continue operating in the same way. It was a very unsure time, and so to risk mitigate, we did open up our own R&D team to support some R&D work that came through. Vigil was the first group outside of China to actually be able to isolate the virus and grow it. And they were fantastic and still are fantastic for supplying 360 with the various variants that are going through. I think BA5 is the one that we've just secured in our lab and are working into a micronute assay. And then we started to announce in March that we did have the virus and would be able to support R&D work in the COVID space. We continued doing this through April, and then this happened. We got this absolute flood of interest coming into 360 Biolabs for R&D work. And these were very, very unusual pieces of work. Some of them were herbal extracts and seeing if there was antiviral activity against SARS-CoV-2. There was hand sanitizers, you know, Australian garlic producers. They were adamant that garlic was going to cure COVID. A range of different masks. You know, the one on the right-hand side is probably the funniest story. There was something that was going to cleanse the air, essentially. And when you're talking to someone who's got a product, but someone that doesn't understand the science, it's very interesting when they explain to you how the experiment needs to work. So what you need to do in your PC3 lab is aerosolize the virus. And then I can come in with my ionizer and I can cleanse the air, and then I've found the solution. And so as you talk them through, okay, so you want my team to be exposed to aerosolized COVID, and when none of us had it at the time. And then how's this experiment actually going to plan out? And they'd sit there scratching their head going, oh yeah, good point. Okay, we can't really do this, can we? So it was an interesting few months of education of the general public who just really didn't understand science, but knew that they had this product that they thought was going to cure COVID. We did have some really great experiences though. This was a good one from the Colin team. So they had a fiber that they found was antiviral. So it did neutralize the virus on contact. So that was a nice experiment and project for us to do. And they then moved off and built that fabric into some masks. And I think they moved them into the hospital system. So that was a great experience for us. And then there's also the Viralese that's now available around the globe. And that's from the Star Pharma team. I mean, that's a repurposed drug, and we saw a lot of repurposed drugs over the time as well. But this was certainly a great opportunity to get a product on the market. And then I'm going to move on to the reason why we're here today and introduce Professor Derek. Oh my God, I've just gone blank. Derek Richard, who's going to present the CARP study. We had a great experience in being able to support this study, but the science is really Derek. So I'll hand over to Derek to present.
Thank you. Okay, I'm going to present a little bit about the project to a point that we engaged 360, and then I'm going to tell you a little bit about that afterwards. So our drug of interest, now our team is interested in genetic stability and using that as a therapeutic target in cancer, which is nothing new. You know, where cisplatin, ionizing radiation, they all target the same thing, but we're trying to target in a more specific way. So we developed a drug, DKLSO2. We went through GLP toxin and rodents and dogs. We had really good effects in cancer, ovarian, lung, and prostate cancer. And so it's in vials, ready to go. And this is what the drug looks like. The target is actually ubiquitous. I'm a microbiologist by training, and I discovered this family of proteins in a volcano in Iceland, first of all, in an archaea called Cephalbus sultaris. And then when I went searching for anything else that had this gene, I found that humans had 2 copies of it. And human SSB1 is the primary target of DKLSO2. So it senses DNA damage in cells. Our normal cells don't have much DNA damage, but our cancer cells do, and that's what SSB does. So how did that go from there? So I'm a microbiologist anyway by training, but how did they go from there back into SARS and other viruses? And the actual answer is the fact that this SSB family of proteins that we first identified in Crenna archaea are actually in all forms of life. And there was being a Scotsman, I was drinking whiskey one night and just thinking about things about the lab, and then I thought, wonder if SARS-2 has human SSB1, because I knew some of the viruses did, and lo and behold, it did. It's a protein called NSP9 in the SARS, and the great thing about it was that by computational look, the OB fold was almost identical to the human OB fold structurally. So we thought, right, let's have a try and see if our protein. So what we do know is NSP9 is absolutely essential for the viral replication process. It just can't survive without that. So we started off doing the biophysical studies in our lab, and we were able to show that, yep, DKL02, our drug, was able to bind to NSP9, purified NSP9, and we saw really strong binding with a KD of around 320 nanomolar. So that was good. We also repeated this using SPR, and we got kind of the same results. And so we're now in a position where we want to push it a bit forward. This was just a trial, a few experiments to see if it was worth carrying on. And that kind of did. So we then fed it into, we've got quite a large team upstairs. So we fed it to a molecular team, and they did a molecular dynamic simulation of DKL02 bound to the SARS NSP9 protein. The interesting thing about NSP9 is it's identical in all the coronaviruses. It hasn't mutated, kind of telling you, one, it's essential for the viral, and two, that it probably evolved early on in the evolution of the virus, the coronavirus family, and thus it kept its structure all the way through. So that's where we got to. And so the next thing was, well, all right, we actually run a cancer lab now, not or an aging and cancer lab. We don't do viral work, so it was a bit out of our area. And so rather than trying to learn the technology ourselves, to get ourselves trained up to PC3 labs, et cetera, because there wasn't much in the way of collaboration capacity. So what we did was we went to a gentleman called Mark Reed, Dr. Mark Reed. He's based in Brisbane. He actually gave a talk here last week, and he guided us to 360 biolabs. And so we then started a collaboration. It's it's one slide which is going to demonstrate a whole pile of work that Angela did and our team did, but effectively, out of this was that the drug was pretty effective against stopping viral SARS-CoV-2 replication, which was great. And so people often ask me, how much does it cost? And because it can be expensive, but the most expensive thing in science is time. Time is super expensive, especially if you're trying to react to something. So we think it was money well spent. And basically, these are just the growth curves for the expansion of the virus. You can see here we get in the nanomole range, we get a CC50 of, I can't remember what it is now, but about 30 nanomole or something. So yeah, so that's probably about all I'm going to talk about. The data we got from 360 was really good. It allowed us to give you a full report. So it's not just like an experiment with a sheet of paper. You get a full report. They've covered everything. It's an ISO accredited laboratory, so you're getting quality. And why is that important? So if you're moving to the next step in development, we've got a drug in vials ready going to patients. We're just trying to raise some money to do that. And there weren't many patients for a long time, but now there seems to be a lot of patients that we can in the hospitals just now. But the reason you use a CRO is, well, number one is time is money to me and our lab. So we wanted to push it. We only had a certain time gap to get this working. The CROs also give real confidence. If you're looking for investors, engaging a CRO, it's an independent lab. There's a lot of bad news out there about companies unable to replicate other people's academics' experiments. And that's probably in a lot part because academics really don't do our experiments to GLP standards. So it's often quite hard to replicate everything. And so this basically engaging with 360 Biolabs gave us a document that we could easily hand to the government, hand to the investors, and move forward from there. So yeah, I thoroughly recommend using CROs. And if 360 is in your area, then we had a really great collaboration. And I'll hand back now.
And I'm really just going to finish the story. So I think the CARP study was probably one of the last ones that we actually did do, because what was happening in the clinical trial sector is it was business as usual. We were getting studies coming into Australia still. There was recruitment going on. All the phase 1 units in Australia were still recruiting as usual. So we ended up closing back down the R&D lab again, refocusing on clinical, and we were able to support Novavax clinical trial, as I mentioned before, the first COVID study that was done in Australia, and then followed up by the Clover Biopharmaceuticals team, which was a Chinese group who conducted the second COVID clinical trial in Australia. And there's still a lot of COVID vaccine studies going on in Australia. So yeah, Australia is still attracting a lot of world-class biotech. And that's the end of our presentation. So thank you for listening.
That was really fantastic. So now we have last two speakers, two Andrews actually. So one is Dr. Andrew Masel and Dr. Andrew Prowse. So first Andrew Masel will be speaking. So Andrew is the acting general manager of QGEN Cell Therapeutics, as well as histology facility manager. And Andrew worked before for Queensland Health as a senior scientist, and he also spent 13 years in applied biosystem. Andrew received his PhD in molecular biology from UQ in 1992 and completed 2 postdocs at UQ, one in plant pathology and the second at Equine Research Facility. And the second speaker, our last speaker, is Dr. Andrew Prowse. And Andrew Prowse is a research fellow at AIB and UQ and STEM core facility manager as well. And Andrew has over 15 year experience in pluripotent stem cell or PSC biology, bioprocess, project management, and commercialization. Andrew received his PhD in 2008 from University of New South Wales. And after finishing his postdoc at AIB, Andrew moved to Toronto, Canada to take a position as a project manager at the Center for Commercialization of Regenerative Medicine, where Andrew focused on developing scalable PSC expansion and differentiation to cardiomyocytes with bioreactor technology. And upon returning to Australia and AIB, Andrew continued his work on bioreactor-based expansion of PSCs and differentiation to cardiomyocytes. And Andrew is now focused on PSCs used in phase 1 clinical trials for the treatment of cardiovascular disease. So first, please welcome both Andrews.
Thank you all. Thank you for the invitation. Welcome aboard. So QGEN. So we're part of QMR Berghoff's scientific services support team, obviously collaborating with all our internal researchers and industry partners to try to get what your idea is, get it into a clinical trial. We're obviously growing in the process of either doing an upgrade or a new facility for the Institute. So we're obviously supporting both local and international markets. So the main part is, how do we get your discovery into a clinical trial? Okay. So you can see the middle two is what we look after. You have the research side of it. So you've got your new discovery. So you've obviously validated that in the lab. It's preclinical, very small scale, basic processing, manual, what we call research grade components. How do we get that into a GMP facility? And we can help you with that, okay? So Andrew's going to actually talk about a real world example where exactly that. So he has an idea, he wants to see, can he get that into a GMP process? And we can help you with that process. You come and talk to us and we take you through everything. The main part of it is the QC/QA. it's a big part of getting your particular project into it. The regulatory effects go through the roof. You need more and more paperwork to get all that done and get those products used for clinical trials. And then we can also talk about the scale up, okay? Getting that into potential phase 2, phase 3 clinical trials. And then obviously the final goal is, if you want to go global, get that into a commercial field. So we've been around since 2002. We're 20 years old this year. So we've been doing this a long time, mainly with the autologous and allogeneic immunotherapies, obviously with your stem cells, T cells, and NK cells. We've now treated over, I think, 400 CAR T patients. We just clocked over the other day. So we have on-site accredited quality control. Like I said, that's a big part of what we do. Looking at all your sterility, your mycoplasma testing, environmental monitoring, a lot of flow cytometry, and then obviously potency testing and endotoxin. Around all of that is obviously the TGA licensing. We got our license in 2015 for the manufacture of T cells for phase 1, 2, and 3 clinical trials. We can also manufacture other clinical therapies for phase 1, and we can also do other therapies for phase 2 and phase 3 on application. And obviously GMP quality control testing through all your process. If you need to go to the FDA for your IND application, we can help you with that as well for manufacturing QC for phase 1 and 2. And we've also done phase 1 and 2 in Hong Kong and Canada. And we've also passed 2 European assessments. So that's where the client will come out, look at our facility and see, is it fit for purpose for what they want to do? And we've passed all those tests, multiple TGA inspections with flying colors. So obviously funding is a big challenge. You have an idea, how do you get the funding for that? So the best scheme is from the Therapeutic Innovation Australia. You can get a grant, you can get money to actually help you in this process. They're for domestic researchers, and also QGEN actually has a remit where we are part of the reason we're there is not just for QMR-Berkhofer researchers, but to help anyone in the state to try the system out, see if they can actually get it to work. And we have a discounted scheme to help you with that on the prices. So talking about the scale up, you get to know the exact details of every step in your process. Research grade materials versus GMP grade materials are very different and may not be fit for purpose. So that's just one thing that can cause a lot of stop blocks in your actual processing. How much product do you need? And is this process easy scalable? The regulations with the TGA. So you need to notify them, so your CTNs, notify them that you're thinking of doing a clinical trial, and then actually get your CTAs in, your approvals to do this process. The key point, it's not a simple process, okay? It takes time to get all these things in place. And you can have a look at the clinical trials website. We'll give you a lot of information if you're thinking about starting this. And then think about, where do you want to do it? Do you just want to do it in Australia, or do you want to take it globally? Like FDA, Europe, Japan, there's all different regulations that you need to consider. And then the most important ones, as I've talked a few times, is the raw materials. Consider removing any animal or human source material, no antibiotics, and do your current materials, can you use research or higher grade, that is GMP compatible materials, to move easily through the development phases? We can obviously advise and help with all of those, and we do that all the time. So this is our facility. So we have 13 ISO 7 clean rooms and two 8 PC3 and BC3 containment suites. Every clean room is independent. So your standard clean rooms, there's nothing too funky about them. So we've got a pointer. You can see up just to give you the purples, the clean rooms, okay? Green is the QC, yellow is the admin, and the light purple is where you come in at the top. So you come in through the top, do all your gowning, and then you go into whichever room you're going to do your processes in. And we take you through all that when you come out. Location, obviously, the main point about Australia is very acceptable to the rest of the world with the problems. We've been talking to a lot of international clients that want to diversify. They don't want to do everything in Europe or the US or in Asia. They really want to spread things out to keep everyone happy. So the point of QGEN being, and you guys as well here, is that we're all centralized with the hospital, with the Royal Brisbane Hospital. I'm sure most of you know where QMR Berghofer is, but just in case you don't, we're actually at the back of the Royal Brisbane Hospital precinct. So a couple of case studies. So several years ago, we had a US company looking at developing some T-cell therapies. The lead product was actually in phase 3, and they were actually looking to custom build their own facility. It was actually under construction. So they licensed 2 preclinical assets from us, both autologous and allogeneic. So we originally contracted to manufacture one of these products for phase 1 in the US and Australia, and while they were waiting for obviously a facility to be built and come online. Basically, it was so successful that they've kept us online even today, like 3-4 years later. Even though they've got the new facility built, we're still producing all these CAR T cells for these guys. Because of that diversification, again, that they really like that we're doing a fantastic job. We'll just let them keep doing it, even though it's on a smaller scale than what they can do, and it just keeps their options open. A second case study was an investigator-led trial. So this was a phase 2 trial in Australia and Hong Kong, and it was mainly around what we were controlling was all the recruitment, collection, shipping, manufacture, and release, effectively the supply chain. So we had patient collection in Hong Kong, shipment of blood to us. We would manufacture, QC release, and then reship that frozen product back to the patients in Hong Kong for infusion. So again, a much simpler process, but we can handle everything through your actual process. So I'd also like to introduce Andrew. So Andrew's going to give you, as I said, a real world example of a researcher that had an idea and how can they get that into the actual process. Thanks, Andrew.
Hi, good afternoon, everyone. My name is Andrew Prowse, and as per introduction, I work at the University of Queensland, specifically at the AIBN. Prior to my introduction to QGEN and the TIA Pipeline Accelerator program, my colleague and I, and it was just the two of us, producing cardiomyocytes derived from pluripotent stem cells, and we were passing those cardiomyocytes on to Dr. James Chong at Westmead in Sydney. And he was using a preclinical pig model for myocardial infarct. And at the time, it was going really well. A number of groups around the world were using similar processes and approaches to also get towards phase 1 clinical trials for treatment of cardiovascular disease. And while the animal studies at the time were going well, we thought it would be a good chance to branch out and try and see if this would fly here in Australia in a therapeutic environment, and to see if we could do a pilot study for GMP manufacture. And that's how we came across the TIA program. And as it was, there were only a couple of cell therapy related institutes or organizations as part of that program that we could access. One of them was luckily QGEN. And I can't speak highly enough of this program and its ability to, because, well, I won't lie, access to GMP compliant facilities, the documentation, all the standards and everything. It is A, big learning curve, and B, it's expensive. So anything you can do to leverage that and get in is a great assistance. So we were greatly indebted to the TIA Pipeline Accelerator program. So how did it work for us? What it gave us, aside from a little financial boost to access the facility, we got training in GMP and facility access. We leased a clean room as a result of the funding. We got constant feedback and support from the QGEN team with regards to regulatory advice. They handled all the raw material purchase for our pilot studies, did all the QC and the QA documentation around that. They also contracted them to do the quality control and batch release, which under the pilot study was limited. We didn't have a great desire to do a large amount of batch testing. And they also provided background clearance of the line. So prior to entering QGEN, we had to provide a whole range of documentation around our cell line to show that it was at least suitable for use in a GMP manufacturing facility. As you'd understand, they handle a lot of sensitive material, and we can't be bringing in any old material into the system. This is just a little bit of an overview of our process. By no means, I'm not going to delve into the science of it. I welcome an invite back from TRI if anyone wants to see the whole process. I'm definitely keen to talk about it. And we're getting out there now as a team and promoting what we're doing. But just roughly, it's an approximate 28-day process, start to finish, where we thaw cells from our master cell bank or our working cell bank, expand those briefly for approximately 8 days in 2 dimensions. And then we translate that to a 3D aggregate-based system for a short while before differentiation. And that differentiation is continued in those, and in this case, it's vertical wheel reactors, for a further 15 days before they are dissociated into single cells and cryopreserved. This is the final process as it stood when we went to QGEN. When we originally contacted them, we had a vastly different and much more complex process. Through serendipitous outcomes, be it COVID, other funding issues that we had, etc., our time moving into QGEN was actually delayed. But what that meant was we could seriously think about refining our process, slimming it down to the bare necessities to make it more cost effective, because as I said before, everything you do there is 3-4 times more what it would be in a research setting. So if we can limit it and create our process as simple and as refined as it can be, then that's beneficial for going on to phase 2, 3, where you're going to need to scale up and produce much more cells. And it just makes day-to-day work in the facility itself much smoother and simpler. Without going into too much detail, as I said, QGEN handled the sterility, endotoxin, mycoplasma, and cell count and viability testing at the end of our process for a release process to make sure we've gone through everything in a compliant manner, and then it would, while only a pilot run, in theory be suitable for clinical trials. We then had a number of AIBN or research-based characterization protocols that we would go through in order to validate our final product as well. So yeah, not delving too much into the actual science and what we did, the outcomes of this were far more beneficial than the original cost involved and the time we spent there. So we gained a lot of valuable insight into the operations of the clinical manufacturing facility. My colleague and I were the ones who went into the GMP facility and actually did the manufacture, as opposed to translating the process to QGEN staff, which is another alternative. Some of the things we learned, as I've mentioned before, we triple the estimated time for each manufacturing step. For example, in a lab-based scenario, if you've got just a simple media change in your cells, add to that the process of gowning up, the environmental monitoring you have to do, other QC and documentation filling in as you go along, your batch release criteria, a 15-minute process suddenly becomes 2.5 hours. So if you're already in the lab all day, you can be prepared to have a very, very long day. Sample requirements for batch testing and release assays, a lot of that sort of stuff we were not familiar with. Again, I'll give a very specific example. For sterility testing, it's generally recommended that you use 20% of your end product for the sterility test, which meant we had to significantly change our process and alter it in order to still get the appropriate yields for a clinically relevant product, while at the same time having enough cells on the other side to pass off for batch release and criteria testing. And I've already mentioned the environmental monitoring. As I mentioned earlier, the best part of this was that we were forced to think about every single step of our process and decide whether it was truly necessary or not. And through the advice and the help of the staff at QGEN and our project manager, we're able to pare this back and understand what is really necessary in order to go from thaw to end product. We also established relationships and a path forward now as we've received additional funding to enter phase 1 clinical trials, where we're now starting to talk to QGEN and developing a master cell bank or a working cell bank at their facilities. We have a much greater understanding of the costs involved and a comprehension of the realistic timelines involved to get to the clinic. And we have significantly advanced the skill sets of our team, which now has grown as well as a result of additional funding. So out of this preclinical pilot study, we got a lot of valuable information in order to progress to the phase 1 clinical trial. So that's me done, and I think that's the end of the QGEN component of the talk. Thank you.
So we have time for questions, and you can either type your questions or just ask yourself. So I'll start one question first. So I'm an early mid-career researcher. What advice would you give? Because it seems a lot of work going to the clinical trial from your lab-based study, and do you recommend doing any trainings or something? Because, you know, there's heaps of regulations required going to the clinical trial. So
Yeah, well, I would honestly say that, you know, obviously Andrew and team have been able to learn things along the way, and that's certainly useful for your own knowledge bank. But there's so many great service providers out there that can actually know so much more and can take you forward much faster, that I would suggest that you just identify those service providers that can help you move faster. And you do learn as you go anyway from just working with those service providers, but you can get your projects to the clinic much quicker if you do it that way, I would suggest.
Yeah, totally agree. Yeah.
Yeah, I'll just add to that. There are fellowships you can get that will place you in a commercial lab for a period of time as well. So I think Ready is one of them. And I think that's an access of people moving between academia and industry.
yeah. Any questions? Anyone? Is there any on Zoom? Okay, looks like I'm going to ask the question. Especially Derek, the inhibitor. So because now we have this COVID different variants going and all this. So what's your plan? Like testing, is it going to work for all the variants, and maybe we can get over COVID or not?
I guess like you'd have to be hopeful, because every coronavirus that's ever existed has NSP9 in it, and they're almost identical. You know, we're talking about 99% identity between them. So you'd hope the variants would be just as susceptible as you get. You get a lot of zoonotic ones as well, coronaviruses, not just human. Yeah.
Especially the Omicron.
Yeah.
Are you going to test against Omicron, or?
Does Angela have Omicron?
Trade secret.
Thank you. Thank you to all the speakers for a great informative talks. I'm wondering, in terms of timeline for an assay development and validation, if you get given the compound, how long are you looking at for doing that part?
Yeah, it's a good question. I think industry standards is about 6 to 8 weeks. So that's what you've got to be able to do. So generally we'll develop, you know, our ELISAs or our small molecule PK assays in a 2 to 3 week period, and then validate in a 4 week period. So you have to move quite quick when you're working commercial, as I'm sure Andrew knows. And you have to be able to problem solve very quickly as well. So, you know, that process of knowing what the next experiment is next, you don't go on that search for, you know, we could be doing it 100 different ways. You have a very, very clear set of path of 6 to 8 experiments that you'll do every time that will pretty much ensure that you get an assay developed very quickly.
Yeah, ours is a little different depending on the context of the material. We've just had a recent client who expected 2 to 3 months to do the check transfer, and it's taken 9 because of supply issues and various other constraints. And just as Andrew was saying, their process was quite complicated, and some things just couldn't be done at a GMP facility. So we had to discuss that with them about what we can do more in the process development phase We do all that as well while we're actually discussing it. So it's getting those realistic timelines through to people is probably the hardest thing, right? Everyone wants to do it yesterday, but in reality, in a GMP facility, sometimes it just takes time, and you just have to be aware of that. Yeah. No, that doesn't. We're not commercial, okay? So we're part of QMA Burke office. So even though we have our set prices, we're pretty flexible. So that particular case, we have a set price for the tech transfer and the PD, and that stays. Even though it took longer, there was no additional charges.
That would be a little different for us.
Yeah, exactly. Yeah.
Yeah. So yeah, we would continue putting FTE days on, depending on how much work.
Well, we
is
can
required.
cover that a bit easier. Yeah.
I'll maybe mention we got a quote to do some work with QGEN, and one of the good things is they have a lot of equipment in there. So the cost of developing something can be quite scary when you have to buy your own flow cytometer, etc. Whereas I think QGEN has a lot of equipment in their facilities that you can use.
And we have a discounted price for Queensland researchers. All right. So our commercial prices versus your researchers is very different. But one of our remits at QMA Burke offer is to give you that avenue through to clinical trial. So we want to make it as easy as possible. And we know cost is the biggest issue. So we try and give you some of that back. Plus with the TIA is very good in giving you grants. So.
Yes, I think that applies to both the researchers who are using this. What were the challenges you faced from going your lab-based study to the phase 1 clinical trials, and especially the terms of funding? Like, it's so hard to get funding. So how did you manage? Like,
well, the pipeline accelerator was a good start in order to get the pilot study done and a greater understanding for our team on what the costs were involved. I have to say the cell manufacturing side of things is pretty straightforward in terms of cost. That's not too bad, even if we were to contract it specifically to QGEN and their staff to physically do the work. We're aiming to reduce that cost burden by having our staff do it themselves after training in the facility. But what has been I suppose a relatively rude shock, and we kind of knew, was all the additional testing that's required after that. So I alluded to the master cell banks we're going to be creating soon. There's a lot of additional testing for compliance that needs to be done, and they're going to be sent to external CROs, and that is a large sum involved in just validating that bank for safety and use in clinical trials.
done with question, but thank you everyone, and please welcome. Please join me in congratulating everyone. It's really fantastic talks we had today. It was really amazing. Thank you.